Okay, so what I'll do this morning is I'll present another way that this group, PepPro Analytics and the University of Missouri, is really developed in terms of thinking about a proteomic approach, but I would say a targeted proteomic approach to take a look at endogenous allergens. And so this is a nice presentation after crystals in thinking about the technology itself and, asking the question, can I measure a targeted set of proteins in a complex matrix, which we have in the case of seed. And so PepPro is really an outgrowth from Dr. Jay Thielen's lab at the University of Missouri. And so they haven't been around too long, but even before they started this analytical approach with the mass spectrometry, they are really experts in the field of taking a look at seed proteins. So they've got a long history of expertise in doing that in particular. In fact, they've worked a lot with the DIGE and regular electrophoresis technology. In fact, Jay's got a nice, I think it might still be up, website on all the DIGE, really presentation of the data and characterizing proteins throughout the growth of soybean seed. And so what they wanted to do today was really show you their this bottom-up approach to proteomics techniques, and the PATC has been working with him for quite a number of years now. And really taking a look at the mass spec methodology was really an outgrowth of knowing that the EU regulators were highly interested in having something to measure endogenous allergens with. Also recognizing there was a lot of focus on what was available at the time, which would have been some kind of electrophoretic technique. Two-dimensional electrophoresis was obviously out there in the literature, and so the goal, at least for the PATC, was to take a look at something that might lend itself to multiplex approach, so multiple proteins, had the potential to be quantitative, which has really just been developing quickly here the last few years, and that's what a lot of this is. And so the bottom-up proteomics approach is really taking a look at your targeted proteins that you want to measure. They use a multiple reaction monitoring technique, and then there's the ability to quantitatively measure the concentration of a protein as you have extracted it from the seed. They refer to that as absolute quantitation because you're actually putting units of measure to it. And then, of course, thinking about the peptide design. So in terms of mass spectrometry, not all peptides are created equally, so to speak. Not every peptide that you might want to measure flies well on the mass spectrometry instrumentation. So that's important, and they have expertise in that. So then for allergen quantitation, using the AQUA is a branded name for actually using the labeled peptide that you're going to use to help quantify. So you'll hear AQUA frequently. And really the data that they have to present today is looking at the environmental influence and what it does to concentrations of different allergens. And then the other variable that's been looked at, just as you saw with Crystal's work, when you have different cultivars of a crop such as soybean, does that play a role? So the bottom-up quantitative proteomic technique itself, what you're doing is a purely analytical technique. So unlike really the earlier days of proteomics, so to speak, and with a focus on allergens, where maybe you were doing two-dimensional electrophoresis, then applying serum on top of that so that you could use the serum IgE to bind only the proteins of interest, not every protein in the seed. This is purely analytical, so serum is not involved. And so you can measure the peptides, the intact parent mass of a given peptide, but because this is tandem mass spectrometry, you're going to have a destructive phase, which is the second part of mass spectrometry in their case. And so you have a nice quality control feature here where you're going to get sequence information for each peptide that you've targeted. So every run generates that kind of quality control information, which is a nice advantage of this. Every run is not isolated away from other quality control measures that you would have had to run to verify that you're actually looking at what you say you are. And so the quantitative proteomic method itself is really chromatography dependent quantitation. So you're going to integrate the area under a peak after you separate the proteins by chromatography. But you can also do a method that is chromatography-based or independent, but it's quantitative by spectral counting. The lab's done both of those. And with the first technique there, where you are going to use chromatography and then the tandem mass spec, the whole peptide signal is measured as it elutes. And then you're going to fragment that peptide, and that's the feature of the tandem mass spec. With spectral counting, you're going to count the number of times a protein is identified by the database searching of the spectra that you get. So there's some fairly heavy-duty math that's going on and some bioinformatics where you're verifying that what you see and what you've measured really is what you think it is. And so different technologies, apparently in the literature and in their world of expertise, there's some opinionated landing on one technique versus the other. And he's just highlighting there that we're going to be talking about that. So a graphic here, this is an active graphic, and so what this is intended to show is that you have some peptides and you've got a complex matrix on the left, so you've got more colors than what are shown. How do I make this work, Nancy? Oh, okay. So if these are your targeted peptides here, they're in a complex matrix, and so where they start is they take seeds, grind them up, and they're going to do alcohol-based extraction of all the proteins present. You don't want to selectively leave proteins behind if you can help it. They've done a lot of work in standardizing that part. You heard this morning how important that can be. With the soybean work in particular, they worked really hard and they actually published on nothing but how to extract total protein from seed because it's so important. But anyway, the concept here is you do want to do not broad spectrum-based proteomics. You actually know what you're looking for, and so it's targeted in that sense. And so if these are it, they're going to go through a peptide mass filter. You're going to let certain ones in or pay attention to certain ones. And so they're filtered, so only the desired peptide is analyzed. There's really too many peptides, obviously, in a complex seed matrix to look at all of them. But because it is mass spectrometry, you can pay attention to only certain ones. Then you're going to break the peptide bonds. They're fragmented to provide the amino acid sequence information. And so that final signal is specific for the peptide of interest. Mass matters and gives you the sequence information. They can keep track of that and measure it. And then you do get the signal there at the bottom, which can be quantified. This is happening all very quickly, so there's a lot of setup and setting up their databases, your sequence databases, so that you always know with a particular run what you're looking for. You can measure greater than 10 different peptides every second. So there's the multiplex feature here is pretty incredible. You saw two-dimensional gels, which obviously will separate proteins on a pretty broad scale, and so you can get, what, hundreds, Crystal? Easily hundreds of dots. And so this even expands upon that and thinking about proteomics generally. But again, what we're talking about here is targeted. So then I mentioned the AQUA branded peptide. What that really is if they can find their peptides, they know they fly well on the instrumentation, the next step is to think about, well, taking those best peptides that represent the protein and making effectively standards, which you can then spike into a sample and follow that spiked peptide. Since you know how much a spiked peptide can be in a sample, then you've got something to quantify with. And the AQUA is the brand where they send out the peptide, get it synthesized with the label of interest. There's two different ways they can do that, the two different isotopes. And really what they're doing with this is because they can get this label, they're going to change the mass either by 10 or 8. And that lets it separate by mass on the mass spec, and they can keep track of that peptide separately from the native peptide. And eventually I'll show you how they get that ratio, which gives you the quantification. So they're changing mass with the spike. You know, everything else about the peptide is the same except for the isotope. And just as an example here, they're showing a native peptide here with its mass. And then if they send out and get the AQUA branded peptide labeled the way they want, if it also passes qualification and flies well, then they've got something to act as a standard. What this looks like graphically, they're obviously taking that extract from the seed. It's got the allergens plus everything else that's in that seed. They're going to take that AQUA internal standard and mix it, And because they can separate by either 8 or 10 mass units, that's easily resolvable on their equipment. And you can get the stoichiometry and the integrated ratio there to give you something to quantify with. And so they're showing here an example of near isobaric peptides may not be distinguished during low resolution analysis, but the retention times finalize the peptide ID. So they're showing 1 or 2, and then the aqua is there in the same place, and the difference is going to be the mass. And so right here, they're verifying that the sequence itself is the same, and in this case, the example is showing something that's different by 3 other peptides, and that will separate away from the one of interest. And so I think the point here was just to give you a feel for the selectivity and the specificity in terms of starting with a very complex matrix, but zeroing in on exactly what you're interested in. And so, again, just zeroing in on the one of interest and being able to discriminate, in this case, just a half Dalton difference. So then on to peptide design. I mentioned that thinking about how you're going to design peptides so that you can look for the proteins that you're interested in. As you saw with the 2-dimensional gel, a given protein class or family is going to have many isoforms. If you can't resolve those, you would either need to think about producing a quantified amount of all of them together, or discriminating between those isoforms if they differ in a way that you can discriminate by selecting peptides that do that job for you. So in this case, they've got the glycine subunits shown. Here the box is to show a part of that sequence that's specific only for the G2. And then they were able to find a portion that was specific for G1. So if you had that question, you were interested in certain isoforms or subunits of a mature protein, if the sequence allows for it, you can have peptides, and if they fly well and pass all those other criteria, in this case, you could look for these 2 peptides and tell somebody what the concentrations are of those and resolve them. If your goal is to ignore isoforms, in this case, there's a common one across all 3, and you would just report the value or the concentration of glycine, G1, 2, 3 in parentheses. So the ones they've measured to date in soybean are listed there on the left. They've looked at some peanut and the major corn allergens, see the Zea M14. And so that's just a listing there. You can see the soybean and their work in a couple publications by the folks in their lab. And I'll show some data that they've worked on here recently, including a publication that they've worked on with DuPont. And so the PATCs, like I mentioned, worked with them a lot. Greg and DuPont have worked with them on a project. And really the goal here was in thinking about, okay, if the technique itself worked, what's the first thing you might do about it? So knowing that, potentially the European regulators were interested in that, the idea was not to quickly jump on thinking about how we were going to compare test material and control material. In this case, no one had known before this what the concentration of, say, beta-conglycine and alpha subunit was. Well, the work that made sense was to understand natural variability, so the same kind of work as with the rice. And so these 2 different studies differ in the sense that the samples were sourced from field-grown material, but fairly isolated regionally. So you see most of the sites here in Iowa, 1 in Illinois, 1 in Indiana. The DuPont-Pioneer project was geographically dispersed much more. What they had in common, and I'll show you this, is that the idea was to also look at cultivar differences. So again, like the rice project, it's rice, or in this case soybean, but does the genetic background lend or provide a measure of the variability? So in the Iowa study, and I'm going to blow these graphs up, so don't worry that they're so small on the screen just this minute. I'll get past the graphics here. And so what I'm going to show you when I blow these up are both of these graphs just a bit bigger. But you see the 9 sites here and the 6 sites in the multi-state study. And if you did nothing but use a statistical measure of variability, when you're isolated regionally, the genotype was, this is a negative number here, so the p-value was very significant. If your samples are spread out geographically, the environment accounted for the differences. So here I am zoomed in on the Iowa study, so geographically isolated, and you have a fairly consistent pattern here. Genotype 1, 2, and 3, and the pattern is not that it goes from a lower amount in one to a greater amount, it's that the pattern for blue, red, and green is consistent across each of the sites. So in this sense, being geographically isolated appears to have allowed the ability to measure these proteins, show that genetic variability was the bigger factor in any of these differences. And so total micrograms per milligram of total protein, you're in the range of a low of just under 150 to maybe 180 there, 185.
I'm sorry, Scott, just for
Yeah.
clarification. Those are all those proteins that you showed on the previous slide. It simply says allergens.
No, this is just a selected one, sorry. Yep, he didn't have that marked, I don't believe. No, I'm sorry, this is summed allergen. So one of the things you can do if you do measure the 9 proteins like we did in the PATC work, and same there, you can say, I'm going to ignore the specific allergen because a person doesn't eat one type of allergen from something so complex as soybean. So you've got quantitative numbers there, you can just sum those values. So that's what they did for this presentation. Thank you for catching that. And so to contrast with this, where some very straightforward statistics tell you that the genotype's playing a bigger role in explaining the difference here, with DuPont's study, where you've got samples spread out geographically across the mid and eastern US, you have a significant effect of the environment that overwhelms what difference there may be between cultivars. So now what you can't do is compare this study directly with the previous one. They're not matched samples that way. Really, it's just this is a very beginning or early look at, we're past the technique itself. The technique is very reproducible. This is just to try and understand where the variability lies so that you can understand how you might even begin to think about an experimental design. That's really the purpose here. And so the summary is, PATC and really this group, PepPro Analytics, is taking their bottom-up proteomics methodology to measure or do selective or targeted proteomics, in this case on allergens. They've got a specific way of doing that with the AQUA-branded multiple reaction monitoring. And so it's important, like I mentioned, there's some distinct advantages. You do have the ability to verify with every sample, every run, exactly what you're measuring, which can be important. It's multiplex. You're looking at the peptide at 2 different levels. You've got the whole peptide mass. You can relate the amount of that back to the whole parent protein. So they don't have the detail in this slide deck on how that's done, but there's some pretty straightforward math to do that. You've got the fragment mass with helps with the quality control. And you've got the internal standard and retention times upon which you can base reproducibility measures. So you've got the ability to measure a protein. You can measure isoforms of a protein family, and you can do that down to the single amino acid level, which helps you do that selectivity in your planning ahead of time. So that's what they have for this. Some of the same questions would come up for this work as with rice, and we can talk about that if anybody would like to. The funding here has been through the PATC. They've got the collaboration with DuPont for which you've seen. Again, this is Jay Thielen's lab, and the presenter today who's worked with Jay and in his lab is Dr. Severin Stevenson. And with that, I'll take questions.